Assalamualaikum and hi! In this video presentation, we are going to discuss about Chapter 5, Immune System.
Okay, so today I will be explaining to you about innate immunity. So innate immunity is known as the non-specific immunity. Okay, so humans have a lot of defenses that help us to fight against pathogens that enter our body. So in this slide, I will explain to you about the first line of defenses, which is the external or physical barrier of humans. So in this slide, as you can see, we have three defenses that act as our physical barrier. So the first defenses that I will be explaining to you about is intact skin. So intact skin, it acts as a physical barrier that blocks bacteria, viruses, pathogens from injuring our bodies. So the only way that the microbes could get into our body is by going through the damages or broken skins. Okay, so the next defenses is mucous membranes. Mucous membranes is an epithelial tissue which is lined at the respiratory, digestive, urinary, and reproductive tracts. It produces mucus, a sticky fluid that traps microbes. Okay, so the third defenses is secretion. Secretions, it creates a harsh environment for the pathogens. So basically, secretions, it contains of it likes tears, saliva, sweat, stomach acid, and cilia. Okay, so tears and saliva contains lysozymes, an enzyme that destroy bacteria cell walls. And for the sweat and oil secretions, it makes the skin acidic, which is from pH 3 to 5. So it preventing microbial growth. In our stomach, which is we know as the stomach acid, kills microbes in foods or water. And lastly, for the cilia, which is in our trachea, it trap microbes outward and out of the respiratory tract.
So I am going to talk about the first line of defense. The first one, intact skin, which is also physical barrier. The skin blocks pathogens from entering the body by acting as a strong continuous barrier. Its fatty acids and normal flora also help stop harmful microbes from growing on the surface. The second one, mucus lining in respiratory tract. Mucus traps dust, dirt, and microbes before they can enter the deeper tissue. This prevents harmful particles from reaching sensitive organs like the lung. The third one, cilia action in nasopharynx and trachea. So cilia beats upward to sweep away mucus and trap microbes. This mycociliary escalator removes pathogens from the airway before they can cause infection. The fourth one, lysosome in tears, saliva, and secretions. Lysosome is an enzyme that breaks down bacterial cell walls, killing many bacteria. Tears and saliva also wash away microbes, reducing the chance of infection. Number 5, saliva. As we all know, saliva is an antibacterial enzyme, which is contain an enzyme that kill or weaken bacteria entering through the mouth. It also helps us to wash away food particles that contain microbes can use to grow. Number 6, stomach acid, which is in low pH. The stomach has very acidic conditions that destroy most microbes in food and water. This prevents harmful organisms from entering the intestine. Number 7, rapid pH change in the gut. The digestive tract quickly changes from acidic to alkaline condition. which microbes cannot survive. This pH shift helps prevent microbial colonization. Number 8. Normal flora and commensal bacteria. Good bacteria usually live on the skin and in the gut, competing with harmful microbes for space and nutrients. This makes it harder for pathogens to grow and cause disease. Number 9. Vaginal acidity in females. So the vaginal environment has a naturally low pH that prevents the growth of many harmful microbes. This acidity is maintained by beneficial bacteria that protect against infection. The last and only, flushing by urinary tracts. The flow of urine helps wash microbes out of the urinary tract. This prevents bacteria from settling or multiplying, reducing the risk of UTIs, which is urinary tract infection. Acquired immunity is separated into two types. The first one, humoral immunity, which is antibody mediated, and the number 2 is cell-mediated immunity, which is T cells response. We will go through the first one, which is humoral immunity, alias antibody mediated. So, in humoral immunity, B cells are activated by an antigen and divide to produce plasma cells. These plasma cells release antibodies into blood and tissue fluid to neutralize or destroy pathogens. The second one, cell-mediated immunity, which is T cells response. In cell-mediated immunity, T cells are activated by antigens and multiply into a clones of cytotoxic T cells. These T cells directly attack infected cells or destroy foreign tissue carrying the antigens.
Now, we want to talk about the innate immunity, second lines of defense. Second line's defense against infection depends mainly on phagocytosis, phagocytes type of white blood cells whereby first, engage invading microorganism, second, initiate the inflammatory response. Phagocytic cells. Many pathogens that defeat barrier defense of mammals are engulfed, remove cellular debris and respond to invasion by foreign substance or pathogen. Phagocytes attach to their prey via surface receptor and engulf them, forming a vacuole that fills with a lysosome. Main type of phagocytic cells are neutrophils and macrophages. Natural killer cells or NK cells. Natural killer cells patrol the body and attack virus-infected body cells and cancer cells. They trigger apoptosis in the cells they attack. Natural killer cells do not attack microorganisms directly, but destroy viral-infected body cells. Antimicrobial protein, specifically in interferons. Small proteins released by activated macrophage and lymphocytes. Tissue cell infections with virus. Action of interferons is firstly, activate macrophage and NK cells. These activated cells will then identify and destroy the virus-infected body cells. Secondly, induce nearby uninfected cells to produce substance that inhibit viral replication. Limit the cell-to-cell spread of virus in the body. Inflammatory response, specifically when injured. Firstly, inflammation. Second is a localized tissue response to injury. Thirdly, damage to tissue trigger release of chemical signals that lead to inflammatory response. Lastly, chemical signal histamine. When injured, muscle release via histamine.
Okay, so here we're back, and now I will explain about the lymphocyte cells. So basically, lymphocyte cells is divided by two, which is, as we know, B lymphocytes and T lymphocytes. Basically, we just call it as B cell and T cell. Okay, so now I will be explain to you about the origin of the B lymphocytes. So B cell, it developed and matured in the bone marrow. So The B lymphocyte is basically responsible for the humoral immune response. It is antibody mediated. So when activated, B cells will differentiate into plasma cells and produce antibody. And while they produce the memory of B cells, it gives the long-term immunity. So beside the B cell, you can see the origin of the T cell. So T cell developed in the bone marrow. It's the same as the B cell, but the difference is T cell it mature in the thymus. So T cell is responsible for the cell-mediated immune response. So we have three types of T cell, which is helper T cell, which we call TH cell, that activated B cells and other T cells and macrophages. And the second type is cytotoxic T cell, which is we call as TC cell. It destroyed the virus, infected and cancer cell. And for the last one, which is the memory T cell. So memory T cell, it gives rapid response during future infections. Okay, in this slide, I will explain about the clonal sections of the lymphocytes. So we have 4 clonal sections of the lymphocytes. The first one is the antigen recognitions. So each lymphocyte has its own very unique antigen receptors. So when the pathogens enters our body, only the lymphocyte whose receptor match the antigens will epitope and bind to it. The second one is the activations. So binding of the antigens to receptor activates that specific lymphocytes, and this event it triggers the cells to divide. Moving to the third one, which is we call as proliferation. Basically, it's cloning. So the activated of the lymphocytes that undergoes the rapid mitosis, forming a clone of the identical cells. So this will ensure many cells are available to attack the specific antigens. So this is the last of the clonal selections of the lymphocytes, which is we call as the differentiations into 2 types of cells. So as we know, the differentiation into the 2 types of cells, which is B cells and T cells, basically. So in the B cells, we have 2, which is the plasma cells and B cells. The plasma cells is short-lived effector cells that will produce a large amount of the antibodies, while the memory B cells is a long-lived cell that remember the antigen and respond rapidly during the re-exposure. And for the T cells, we have effector T cells as a helper or cytotoxic T cells and memory T cells. So in the picture below, you can see the clonal sections of the lymphocytes.
Now we will talk about the components of acquired immunity. Number 1 is cell-mediated immune response, and number 2 is humoral immune response. Firstly, cell-mediated immune response involves in activation and clonal selection of T cells, resulting in clone T cells consisting of number 1, helper T cells, which functions in assisting other white blood cells, help B cells become plasma and memory B cells, and also activate T cells and macrophages. Number 2, it also results in cytotoxic T cells, which destroy infected cells and tumor cells, and it is also involved in transplant rejection. And lastly, memory T cells that provides memory of past infection and also faster defense for the next new infection. Next, as for humoral immune response, it involves activation and clonal selection of B cells, resulting in B cells that consist of 1. Plasma cells that produce antibodies that are also known as plasmocytes and effector B cells, and lastly, memory B cells that is ready for the next invasion. Next, we will continue with antibody classes, which are the 5 classes of immunoglobulins. Firstly, we have immunoglobulin M, which is a pentamer. Immunoglobulin M is produced first upon antigen invasion and is very effective in complement activation, and it also promotes neutralization neutralization and agglutination of antigens. Number 2, we have immunoglobulin G, which is a monomer. Immunoglobulin G is a main blood antibody for secondary responses. It is the only immunoglobulin that crosses placenta, and it also promotes opsonization, neutralization, and also agglutination of antigens. Next, we have immunoglobulin A, which is a dimer. Immunoglobulin A tags pathogens for destruction. It is present in secretions such as tears, saliva, mucus, and also breast milk. It also confers passive immunity on nursing infant. Number 4, we have immunoglobulin E, which is a monomer. Immunoglobulin E bind to mast cells and basophils of histamine and other chemicals that can cause allergic reactions. And last but not least, we have immunoglobulin D, that is a monomer. Immunoglobulin D is present on the surface of naive B cells that have not been exposed to antigens. It acts as an antigen receptor in antigen-stimulated proliferation and also differentiation of B cells.
And last but not least, we're going to talk about active versus passive immunization. So let's take a closer look about what is actually active immunity. So, active immunity is when our body develops its own antibodies after exposure to a pathogen. Because the immune system creates memory cells, this protection is actually lifelong. However, it takes time to develop because the immune system must first recognize and learn how to fight the pathogen. There are two types of active immunity, which are natural active immunity and artificial active immunity. So what is actually a natural active immunity? Natural active immunity occurs when a person is naturally exposed to an infectious agent. For instance, when a person is recovering from chickenpox, the immune system will produce its antibodies and memory cells. This will usually result in lifelong protection against the same disease. And for artificial active immunity, it develops through vaccination or immunization. The vaccine contains a harmless or weakened form of the pathogen, and this allows the body to produce antibodies and immunological memory without causing disease. Next one, let's talk about passive immunity, are also defined by ready-made antibodies. So, passive immunity occurs when antibodies are received from another person or organism. It will provide immediate protection because the antibodies are already formed. However, this protection is short-term because no memory cells are produced by the body. Same goes with the active immunity. Passive immunity also contains two types, which are natural passive immunity and also artificial passive immunity. Let's take a closer look to natural passive immunity, shall we? So for natural passive immunity, it happens when antibodies is transferred from a mother to a baby. Let me explain briefly about how this antibody transfer process happens. So first of all, IgG antibodies will cross the placenta before birth and will protect the fetus. And next, after the birth process happens, IgE antibodies in the mother breast milk will help protect the baby's digestive and respiratory system. And next, for the artificial passive immunity, it occurs when antibodies are injected to a non-immune person. The examples include anti-venom, antitoxins, and monoclonal antibodies. This method is used in emergency situations and will provide instant protection. That is all from us. Thank you for watching. Bye!